Hi and welcome to today's video. Today, let us see some details on mitochondria. Mitochondria, we know it is a double membrane bound organelle which is found in the cytoplasm of eukaryotic cells. Mitochondria, it's singular, we call it as mitochondrion. These are typically tubular or sausage-shaped organelles. And the mitochondria word, mito means thread and chondrion means granule-like. It was first observed by Richard Ortman and the term was coined by Carl Benda. It is known as the powerhouse of the cell because it is responsible for the conversion of nutrients into energy-yielding molecule ATP, adenosine triphosphate, which is the fuel for all cellular activities. The function known as aerobic respiration is the reason mitochondria are frequently referred to as powerhouse of the cell. And what is this aerobic respiration? What all steps are involved in aerobic respiration? Aerobic respiration involves glycolysis, then Krebs cycle and electron transport chain. These three cycles are together called aerobic respiration, or these are the steps involved in aerobic respiration. Coming on to the origin of mitochondria. Mitochondria are derived from bacteria by a process termed as endosymbiosis that we have dealt with in a previous video. You can go and look after my previous videos in which I have given the details on endosymbiosis. It is believed that this mitochondria arose about 2 billion years ago, and how it happened is when a bacterium fused with an archaeal cell or established a symbiotic relationship with a primitive eukaryotic cell. The closest bacterial relative that gave rise to mitochondria are Rickettsia. Rickettsia bacteria are the closest relative to mitochondria. The first person to recognize mitochondria as descendants of endosymbiotic bacteria was Ivan. So this all just an introductory part. And biogenesis. How do these mitochondria form and how are the processes being carried out relating to their formation and reproduction? They grow by importing most of their proteins from the cytoplasm. They import the relevant proteins, their required proteins, from the cytoplasm of the cell and by internal synthesis of some proteins and replication of the genome. Mitochondria divide and fuse with other mitochondria and thus they maintain their number. And this division may be they may undergo both fission and fusion during their biogenesis. The fission and fusion has improved in recent years our understanding, and with the development of certain assays for their study and the identification of proteins required for both events, we now have a better understanding on the mitochondrial fission and fusion. And how does it happen? The balance between fission and fusion is likely a major determinant of their number. Mitochondrial number, length and degree of interaction are based on the balance between fusion and fission. And what is the relation? When fusion becomes more frequent than fission, the mitochondria tend to become more elongated. If fusion is more frequent, then the mitochondria will become more elongated and interconnected. While if fission predominates, the formation of more numerous and distinct mitochondria occurs. If fission occurs more frequently, then the number of mitochondria increases, while if fusion occurs frequently, then the mitochondria will be elongated and interconnected. And regarding the morphology, the size may range from 0.05 to 1 micrometer in diameter. It is just like the size of a bacteria. And the length ranges from 1 to 10 micrometers. It is, I already mentioned, it is a tubular or bean-shaped or sausage-like organelle, and number depends on the type, size and functional state of the cell. On an average, liver cells contain around 1500 mitochondria. Likewise, each type of cell has a different number of mitochondria depending on their functional status. And coming on to the structure of mitochondria. What do we know about the structure of mitochondria? Mitochondria are bounded by two membranes, outer membrane and an inner membrane, and it also has a space in between these outer and inner membrane. That space is called intermembrane space. And the inner membrane has certain infoldings. The membrane is having some infoldings and that are called cristae. And the lumen of mitochondria, or the interior part or the interior region of mitochondria, is called matrix. Both the outer and inner membranes are composed of phospholipid bilayer and proteins, and they have different properties also. Outer and inner membranes have a different property. First, let's see on outer membrane. It is simple phospholipid bilayer and it encloses the mitochondria. This outer membrane contains large number of integral proteins called porins, which allow molecules to freely diffuse from one side of the membrane to the other. Porins pass molecules which are less than 5000 Dalton in size. Ions, nutrient molecules, ATP, ADP, etc. can pass through the outer membrane with ease. So outer membrane is somewhat more permeable, and there they have an integral membrane protein which is called porins. And intermembrane space. Next we have a space which lies between the inner and outer membrane, and that space is called intermembrane space or the perimitochondrial space. It has high proton concentration. High proton concentration is found in the intermembrane space. Because the outer membrane is freely permeable to small molecules, the concentration of some very small molecules such as ions and sugars in the intermembrane space is same as that of the cytosol. And the proteins present there participate in ATP synthesis, but when released into the cytoplasm, they trigger or it is some other reactions. It is coming to the inner membrane. Inner membrane is freely permeable only to oxygen, carbon dioxide and water. The inner mitochondrial membrane contains proteins that perform redox reactions in oxidative phosphorylation, ATP synthase, transport proteins, protein import machinery, mitochondria fusion and fission proteins. Several antipode systems exist allowing exchange of anions between the cytosol and mitochondria. Next structure is cristae. Cristae are nothing but the infoldings of the mitochondrial membrane. Particularly the inner mitochondrial membrane project inside, or they protrude inside the lumen of the mitochondria like finger-like projections, and these are called cristae. Why do they have such infoldings? It is just for increasing the surface area and thereby increasing the ability to produce more ATP. Stalked particles or inner membrane spheres, and these cristae is covered with this inner membrane spheres called stalked particles or knobes or heads. Then the next region is matrix. It is the space which is enclosed by the inner membrane. Gel-like consistency will be there and dense homogeneous properties will be exhibited, and it contains 2/3 of total proteins of mitochondria. The matrix is important in the production of ATP with the aid of the ATP synthase contained in the inner membrane. Major enzymes include enzymes involved in synthesis of nucleic acid and proteins. Thank you. Hope you all enjoyed. Go through the video well and study well also. The remaining portion of or the next part of the portion will be continued in another fresh video. So till then, bye, take care.